We see.
Great. There's two things I wanted to mention. Number one was related to the fact that just before the break, Victor held up a beer to tease me. And it reminded me of a talk I gave at Duke University a long time ago, where the talk happened in the middle of the afternoon and they brought in beer and food and put it on the table next to me. And I was giving my talk and all the students came and took all the beer and pizza away. And I was left with a glass of water. So, and the second thing is, I told this story, you get to read this poem from Margaret Atwood, which I thought is quite a nice poem. It's actually about the brain, and it really is sort of captures some of the fanciful things about the brain. And in some ways, it leads in because my talk is a bit more fanciful than I think some of the other presentations today, with the possible exceptions of Dr. Hawkins' work, which I thought was just phenomenal. In the sense it covers more of a theory than hard data. And I think it's important to kind of balance our discussions over the next day. And so between theory and empirical data, kind of see how we can pull those two together. So a lot of what I'm talking about is sort of captured in this paper that Victor and I published, I guess, last year. And it comes from things that Victor had been working on before he and I started working together on this, having to do with what's called structure flows and manifolds. And the point of the paper and the point of my talk is to really put forward the notion that a lot of what we're measuring when we're measuring the brain and thinking about modeling the brain, for that matter, is a combination of things that it's doing in the moment, but things that it could also do. So it's the actual and the potential. And the potential is something we miss a lot, I think, in empirical work, because we may have somebody doing a task where they're pressing a button while seeing something on a screen, for instance. But that's embedded in a rich repertoire of other things that the person could be doing, like eating, for example, or drinking beer, or other ways of doing that task. So it moves us from thinking about a deterministic system to one that actually has the capacity to do things slightly differently. And that comes into the idea of complex adaptive systems. Dimensionality has been mentioned, capacity, and so on. All these features kind of come together when you start thinking about trying to represent the brain and networks as a series of manifolds, which are essentially, you can think of them as sort of rules that govern how things can be configured, and the flows, which are actually the instantiations of those behaviors. So I'll try and cover these points through my 20-minute talk. We'll see how we do. The idea of rules in the paper, at least, the first thing to talk about is actually the notion that running has a series of rules. The rules are relatively simple. You put one foot in front of the other, and then the next one, you hope you don't fall on your face. But the way the rules are enacted can differ quite dramatically depending on the context. So you've got, illustration of, Forrest Gump over here running. You've got two young people running up a hill here. You've got two older people standing over the hills around Marseille. And you've got a picture of me back here running on the ice yesterday in Toronto. The rules for doing this are pretty much the same, but how they are enacted in the given situation can vary. And that's the idea behind this idea of trying to measure manifolds, and is that if we can identify what are the rules that govern the expression of network dynamics and then therefore the ensuing behavior, that might help us better understand how these rules are built, but then understand both what actually happens in the situation in that particular case, but also what could happen in a given situation. So it's a combination, again, of actual and what the potential is for the system. And the potential part really comes from characterizing these rules, what could happen. There's another analogy which is helpful in thinking about this, and that comes to do with how you could learn to play chess. Now, how do you learn the rules of chess? One way to do it, and this is sort of the experimental way to do it, was to observe people playing chess. You can measure the moves they make. You can manipulate things, for example, move things in a particular way and see if that's a legal move or not, and then make a large catalog of all these moves that you had done. And that's doable, and that's certainly most of what experimental neuroscience is doing, is they're trying to catalog all the things that happen when you poke and prod the brain in certain different ways. The problem with that is it will take you a long time to figure it out, because even though there's only a handful of rules in chess, the number of potential moves is astronomical. So to really catalog all those things would take an inordinate amount of time. Another way to do it is to model, identify what the potential rules are, and then enact these rules and see how the repertoire is captured. And that's really how we typically learn chess, is that we were given the set of rules, and then we start playing with, how do we enact these rules? Does this actually work? Can I beat my uncle in chess? It never happened, by the way. But can I do that? If I know the rules, can I enact them in different ways so that I can be a good chess player? So, and from the paper that we wrote, one of the quotes was that we understand chess by understanding the rules of play, and understand it deeply by using these rules to build coordination motifs. And you can translate that to how we might try and understand the link between brain and behavior, that the goal for understanding brain and behavior is to determine the rules that govern the emergence of behavior from the coordination of brain dynamics. So the first thing is can we actually build models that embody this concept? Can we find ways to link things up so that we can look at the configurations? And this doesn't actually capture the fact that there's a temporal and spatial aspect to it, but the notion is that we have flows that are constrained by a manifold, the manifold that being the expression of the rules that govern how a system can transition between different states. And yeah, we can do that. We've seen some examples already today of how this can be done in modeling. One of the platforms that we have developed that you probably already heard of is the virtual brain, and there's the people who are guilty of putting it together down here. The platform was built to try and capture large-scale network dynamics. And it's done some good things for us. And I mentioned that we've been able to, for example, I mentioned in the context of Alain's talk, that we've been able to use this platform to see if we add noise to a relatively deterministic system, do we get something interesting? And the answer is yes, we do. In fact, we can reproduce some very nice intrinsic dynamics that look like resting state networks that are measured now in human neuroimaging. And that just comes from taking the architecture, that being the tractography that constrains, provides the structural constraints for the system, and then add dynamics using some sort of mean field approximation. It could be oscillators, it could be biophysical models, it could be phenomenological models, it doesn't really matter. But the notion being there is that the dynamics add that extra kinetic energy, if you will, to the system, and then allows the emergence of some simulated data. And you can use that then to fit back to empirical data. So a lot of the stuff that we have done has been generating bold fMRI, for example, looking at correlations of signals or functional connections, and then optimizing the parameters to fit the networks. And in doing so, you are able to start reconstructing these manifolds that you can measure and you can model as well. And this is some work that actually came from Victor's lab, and we mentioned it in our position paper as well, that by modeling these systems and incorporating the right amount of nonlinearities and noise, you start producing these exploratory behaviors, these manifold explorations going between different states, for example. And that's captured here in these functional connectivity or dynamic functional connectivity diagrams. We've got a change in the configuration of networks across time. And that's reproduced quite nicely in the empirical data, and you can also simulate that. And that's can be done in the virtual brain. And you see examples of this now all over the place in neuroimaging. As a matter of fact, the word manifolds now become the new bad word in neuroimaging, is that as people say manifolds, and they go, oh my God, you've done manifolds. But they have been measured now in fMRI, they have been related to cognitive function empirically. So you can show that in a broad space, relatively low dimensional still, you can show the trajectories are visited that correspond to putative cognitive functions. So yeah, we can model this and give some idea of at least manifolds that are associated with what you actually see. What we're still missing, though, is that notion that there could be things that you don't see, sort of hidden repertoires that are there. So how do we get a handle on that? Tougher, but I think this is where we actually start using the model in a more exploratory way. So we use the model to decide, okay, we're capturing empirical phenomena. And now if we sort of push the model around in the modeling platform, do we start seeing things that weren't necessarily part of the model initially, but actually suggest that there are hidden repertoires that could have implications for how we think about how the system is configured. And this first came from some work that came out of Victor's lab doing epilepsy. I'm not going to go through this slide. This just basically shows you that there's a whole host of work being done that maybe Victor might mention or not, or he probably told you guys already about it. But using the virtual brain in the context of epilepsy and in the context of actually looking at a clinical trial. The heart of that clinical trial is actually this thing called the Epilepter model. And the Epilepter model is a beautiful example of using a phenomenological model to capture a biophysical phenomena. So here you've got a nice example of a single neuron in the rat hippocampus, and it's showing seizure activity. This is pretty much the same kind of pattern you'd see whether it was a mouse or a rat or a monkey or a man. You can model this phenomenologically by the interaction between slow and fast variables that Victor has shown quite nicely. So we've got a slow variable called Z or Z, depending on where you're from, that is a permissive variable that allows the seizures to take place. And as it becomes more negative, the seizures quiet out, and then it rises again, becomes positive, and the bifurcation happens, and you have another seizure going forward. So it's a very nice example of a phenomenology to allow the interactions between slow and fast variables to show this seizure activity. Shown into a phase plot, you've got the change in the slow variable here, going through rest to entry bifurcation, spiral activity for the spiking activity, and then exit bifurcation again, cycling back through quite nicely. And this Epilepter model has been used, as I mentioned, in modeling epilepsy, and that's been at the heart of the clinical trial. So we have a model now. So what else can we learn about this? Can we actually explore more of what happens when you have this kind of configuration in the system that's representing a particular phenomena? So again, Kenza, who's I think still in Victor's lab, did a more broader exploration of the parameters and the behaviors of the Epilepter model. So as you modify the slow variable, again, you have this nice sort of behavior. You got the slow variable changing, you got spiking activity, and then going back to rest. As you start changing the slow variable, you have a separatrix, which puts you on a different part of a manifold, and have quite a different behavior emerging, which is a slow sort of oscillatory phenomena. From a that was not part of the model initially, that wasn't envisioned, but that actually emerged from just actually modifying the parameters to get this nice slow oscillatory phenomena. When they showed this to the resident epileptologist, they said that's actually status epilepticus, refractory status epilepticus. So, and suggests that if you clinically, clinically, if you get in the status epilepticus, that's actually a bad thing. Usually at least to death. And the way it's typically treated is by giving a lot of drugs to try and keep that suppressed even more. And that's actually what the model suggests. That's not a good thing to do. You want to make it more excitable, so push it back up over the separatrix to get it back into there. And that was validated in a mouse model. There's some additional work to be done on that. What this does suggest, more generally speaking, though, is that there is a repertoire that's inherent in this model that may not be there or be actualized, but is a potential. And there's two aspects to this. First of all mammalian brains, at least, can show seizure activity. So showing a seizure is something that's inherent in all vertebrate brains. And also in the same set of parameters that underlie seizures, you also have this potential for having this status epilepticus, which is a hidden repertoire. Fortunately, we typically don't visit that, which would be a bad thing to do. But it is part of, it's the nature of the configuration of our brains that this thing could happen. So it's a configuration that's there. It's part of the way the brain is organized, but we typically don't visit that, which is actually good. So I said epilepsy is a natural repertoire of any vertebrate brain. We don't actually know what the factors are yet that constrain whether that is expressed, and that's obviously a big area of research to figure out how to not allow our brains to become seizure. The question now becomes, can we think about other pathologies as an expression of hidden repertoires? And I don't have an answer for that yet, whether that's a yes or a no. The brain is a complex system. We've heard that many times today. So it could be the case that the pathology that we see can be at least one of two possibilities. One is the primary loss of a manifold, for example, in stroke, where there's an entire chunk of tissue gone. But it could also be a maladaptive response to a primary loss as well, because the system does adapt. It adapts very quickly to changes in its configuration. So hidden repertoires could be unearthed, for example, by the loss of a tissue as well. One of the things that happens, of course, is as the brain evolves and grows up, things develop. Alain mentioned, and this is often seen as well, is that in the infant brain, the system is fairly deterministic in some respects, a very low repertoire. As we grow up, as we encounter things, our systems change. And do these hidden repertoires tend to emerge as we develop? And can we use that then to understand things like risk for decline and so on, and the potential for the system to adapt in the face of damage or disease? This work goes back almost 20 years. This is the heydays of, or the halcyon days, I would say, of neuroimaging, where we didn't know what we were doing, but we thought we did. So we tried everything. And what we found was that you have situations where you start comparing groups. And in some cases, you find that, for example, in old people, they don't remember as well. So you have them do a memory task, and you find that the activation patterns are different than young people. That's not surprising because they're not doing the task. The question though becomes, if you have them doing the task where there's no difference in behavior, is it the same system? Are we seeing situations where you've got different kinds of configurations that could support putatively the same function? This was a task we had done a while ago, just a short-term memory task, and found that in both healthy young and healthy old subjects, that there was no difference in performance in these tasks, but the functional networks that supported behavior were quite different. So you've got the core system here for the young subject, which is more an anterior posterior system supporting visual memory. In the older subjects, this system shows less functional connectivity and instead recruits new areas, frontal and hippocampal, that seem to be critical for supporting that. And the degree to which they show this pattern in the older subjects relates to how well their memory is working in this particular task. So it is a common observation in aging that there are changes in functional architecture, and some of these things may preserve cognitive function. And the question is that an example of a hidden repertoire or an adaptation or some combination thereof? This is an unknown answer, but at least it can be answered now by having this framework where we're looking at not just what the system is doing, but what it could do as well. Another example of changes in aging, there's a very nice paper from Joanna Cabral et al. Looking at, this is called LIDA, leading eigenvector analysis, looking at the change in states, functional connectivity states across time, and actually modeling the progression of the change, and finding that the nature of the progression actually underlies whether somebody, or at least correlates with, I should say, whether somebody shows relatively healthy cognition or less healthy cognition. So good in green is the progression. The ones who show bad, relatively bad cognition were in black. Some work from Damien Battaglia with Victor and I. This is also complementary, just showing again that the progression of functional connectivity, the dynamics, which is over here. I'm not going to go through the slide because it's quite busy. You've got a lot of switching, less switching in this middle-aged subject, and very little switching in the older subject. And that switching behavior actually correlates with neuropsych measures again. So less switching, you have less high cognitive status. So these all suggest that there might be different configurations that could be underlying, for example, a cognitive function. And some of them may have cognitive consequences, some of them may not. This last slide, before I go to the conclusion, kind of gets back to some stuff that Jennifer showed earlier today, and also we talked about briefly with Alain in terms of looking at the measures of, in this case, actually entropy in EEG signals on different age groups. And here, the measure is called multiscale entropy, which looks at entropy, sample entropy as a function of time scale. So we just do, the fine scale is basically the original signal, and then we just do averages. And at each point we average successive points, you get progressively changes in entropy. In adults, you show this characteristic curve where you've got sort of highest entropy and sort of middle scales. If you try and relate that to space and time, that's roughly in sort of mid-range connections. For EEG, it's roughly the beta range for the EEG signal. If you look at kids, and this is true for infants all the way from kids to about 12 years old, the whole curve is just shifted downwards. So there's less entropy, less information across all scales of the brain in kids. Interestingly, in healthy adults, older adults, you have higher entropy at finer scales and less entropy at coarse scales. And that roughly corresponds to more entropy for local interactions. This is amongst adjacent regions, and less for distributed interactions. If you actually look at mutual information, for example, you find mutual information between distal points goes down in healthy aging. And finally, older adults at risk. These are ones who showed cognitive decline that transition to dementia afterwards. They do not show the increase in fine scale entropy and actually show higher entropy at coarse scales, and that's actually predictive of cognitive decline. So again, it's suggesting that there are configurations that go across space and time. This is captured now, and we call it noise, we call it entropy, wherever you want, that relates to the integrity of cognitive function and may in fact predict changes in cognitive function in the case of elder subjects at risk. It may predict, for example, the lack of a particular repertoire that's necessary to preserve cognition in the face of damage or disease. So this gets back to the first slide again that I mentioned earlier, that the notion that we're trying to put forward is that we're trying to think about how to model the brain, and that captures not just what the thing's doing, that thing being the brain, when we're measuring it, but also what it's doing when we're not measuring it. So what is the potential? What are the possibilities? How does it relate to dimensionality? And that this potentiality could relate to the capacity of the system, both in terms of its cognitive capacity at the moment, but also its capacity to adapt to change, whether it be development, healthy aging, or disease. So I'll leave you with the poem and be happy to take any questions you have. Thank you.